and mortal taste sweet. Okay, metabolic pathway. Metabolic pathway and enzymes. Metabolic pathway link reaction, okay, reactant and product. The truth is that the reactant, let's say A, after the first enzyme catalyzes the reaction to become product B, and then product B will become the reactant of another reaction, so that another enzyme can convert product B as the reactant to become product C. And then product C will become a reactant for another reaction, so that the enzyme 3 can convert the product C or the reactant C into product D. In other words, the product of one reaction very likely is the reactant of next reaction. We call this metabolic pathway. They are linked together. It happens in our body very, very often. The reaction in our body is not just a one-step process, not just okay from A to B catalyzed by enzyme 1 and that's it. No. Usually it's a chain of reaction. And not only that, usually it has a lot of branches. C may go to a D prime, and D prime may go to E prime, and so on and so forth. So you have side branches, you have different branches, okay? It's not as simple as or just a strict chain of metabolic pathway as shown in here. In reality, all these reaction pathways, they are very complicated. And then we talk about protein hormones. Protein hormones, we gave you the example insulin and growth hormones. Insulin regulates blood sugar level, specifically lower glucose in the blood. So it decreases your blood glucose level, and insulin produced in the pancreas. Remember, well, we talk about two different types of hormone, protein hormone and steroid hormone. Let me ask you this question. For protein hormone, does it require a cell membrane receptor, or can it go through the cell membrane and binds to a cytosolic receptor or nuclear receptor? So question is, protein hormone receptor, is it located on the cell membrane, cytoplasm, or nuclear plasm? So where is the location of protein hormone receptor? I'll give you 5 seconds to think about this. Okay, time's up. Yes, you can find protein hormone receptor on the cell membrane surface. So insulin, you have a insulin receptor on the cell membrane surface. When insulin binds to the cell membrane surface receptor, what will happen? Then you will tell the cell to absorb glucose from the blood into the cytoplasm of the cell, so that the blood glucose level will be lowered, and the cell can also convert glucose into glycogen for future storage. Growth hormone, as the name suggests, it enhances growth in the bones, in the muscle, by cell division. Specifically, the cell division here we talk about in the bones and in the muscle, we call it a mitosis. It is produced by the pituitary glands. Specifically, I want to say that it should be called anterior pituitary. Anterior pituitary. And the anterior pituitary gland is under the control of something called hypothalamus. Hypothalamus will secrete something called G growth hormone releasing hormone, GHRH. GHRH, growth hormone releasing hormone, will bind to anterior pituitary glands. When GHRH bind to anterior pituitary gland, the anterior pituitary gland will produce growth hormone to go to the bloodstream. Blood protein, yes, protein forms something called hemoglobin or antibody. They are both hemoglobin and antibody are considered as blood protein. So as you can see that this picture is a lot of red blood cell. Inside, as you know that you may have known that the function of red blood cell is to carry oxygen. Some of you may even know that the function of red blood is to carry both oxygen and carbon dioxide. How do red blood cells carry oxygen and carbon dioxide? Inside red blood cells, they have millions of something called hemoglobin. Hemoglobin, they are quaternary protein structure. We'll take a look at them right now. So you can see that, hemoglobin, they have 2 alpha chain, and they have 2 beta chain. And then in each alpha and beta chain, they have heme group. In other words, they have 4 heme group. So 1 here, 1 here, 1 in here, and 1 in here. The heme group, in the center of a heme group, it has an iron ion. It's an iron ion. It is actually an Fe2 plus ion. And it is the heme group that is responsible to carry oxygen or carbon dioxide. So the hemoglobin contains the red blood cell is made up of 90% of the hemoglobin. The reason is that red blood cell contains millions of hemoglobin molecules. I don't require you to memorize 270 million, but you can just estimate just millions. Because of that, most of the red blood cell, they are hemoglobin. Red blood cell, they do not have nucleus. And then function of the hemoglobin you need to memorize to carry oxygen and CO2. Very important. We have a 4 polypeptide chain. That's why the quaternary structure has 4 tertiary structure, and then it has 4 heme groups. Each tertiary structure has a heme group, and there's a spelling mistake here. For heme, H-E-M-E, and the groups, you can put an S in here. Heme group, there are the Fe2 plus ion. Okay, sickle cell anemia. What happens if something goes wrong with your blood cell? For example, if you have a single mutation in your in the sequence of your DNA from CAC into from CTC into CAC, single nucleotide mutation will be good enough to give you sickle cell anemia, because when you change one amino acid, when you change one nucleotide, you change the entire amino acid from glutamate acid into valine. What happened is that two things happen. One is that the red blood cell will become sickle shape. The red blood cell will become sickle shape. Second thing is that the amino acid will become non-functional. The reason is that now after you change glutamate acid into valine, then the 3D structure of the each polypeptide of each tertiary structure will be changed. When the 3D structure is changed, then even though they contain the heme group, they may not be able to bind the oxygen easily. Okay, last one, transport protein. Protein acts as a transporter to transport different substances across the cell membrane. For example, we have something called carrier protein and channel proteins. Usually channel proteins transport charged ions across the membrane. For example, sodium ion, potassium ion, these are the ions that require channel protein. Why? Ion, they are charged, they are hydrophilic. And the cell membrane, they are usually phospholipid bilayer. In the center of it, you have hydrocarbon tail or fatty acid chain. These are hydrophobic. These are hydrophobic fatty acid chain. Because of that, charged ion, hydrophilic charged ion cannot go through this hydrophobic fatty acid chain. So that's why they need a membrane protein to allow them to go through it. And carrier usually transport large molecule. So what would be the good example of a large molecule? In this case, would be glucose. Glucose is a large molecule that require a protein carrier on the cell membrane to help them to go into the cell. Remember previously we mentioned about insulin, yes? Insulin, as I told you that after insulin binds to the cell surface membrane receptor. After insulin binds to the membrane receptor, then it will trigger a G protein coupled cascade. And then what happens is that the one of the many changes that of the insulin, this hormone cause to the cell is that it will increase the expression of glucose carrier on the cell surface membrane. It will increase the expression of carrier protein on the cell membrane, so that when you have more glucose carrier on the cell surface membrane, then the cell can absorb more glucose into the cell. Then it will help to lower the blood glucose level. Last slides, we will talk about something called denaturing of the protein. Denaturation, meaning that the structure of the protein is being disrupted or destroyed. In this case, okay, here folded protein here. This is the what level of organization is this folded protein? Do you know what level? Let me give you 5 seconds to think about this. Okay, this is a tertiary structure. Tertiary structure. After denaturation, then the tertiary structure can become a primary structure. How do we denature protein? By heat, extreme pH, acidosis, alkalosis. Acidosis meaning that your blood becomes too acidic. Alkalosis meaning that your blood becomes too basic. So remember I told you that when you go to a hair salon for ironing your hair or for perming your hair, it is exactly the same thing. You apply high heat for ironing your hair. You expose your hair to extreme chemical so that you can change the pH of the hair. Then you can denature the protein. Or the presence of certain heavy metal, lead and mercury. That's the reason why lead and mercury, they are poisonous. They can affect the protein structure. Interesting. Denaturation, usually it is irreversible. Meaning once you damage the protein, it cannot go back. It cannot automatically fall back into the original shape. So that, one real life example of denaturing protein other than perming your hair or ironing your hair is basically cooking. Yeah, you do this every day when you cook. What do? You heat the food. Why do you cook? Well, for some of my students may say that, to make the food taste better, and then some of the students will tell me, to sterilize the food, yes, to kill the bacteria on the food. How do we kill the bacteria? When we heat the food, bacteria on the surface of the food, they will also receive heat. What happens is that during high heat, the enzyme, they are made of protein. The enzyme living inside the bacteria, in the cytoplasm, inside bacteria, they will be denatured. When the enzyme in the bacteria denatured, then a lot of chemical processes in the bacteria cannot be carried out. When this happens, the bacteria cannot carry out the normal chemical process in their body, then they will die. So that's how we kill the bacteria. That's why we need to cook our food thoroughly in order to sterilize the food. So that's the end of this lecture.